Epilepsy, a condition affecting millions worldwide, has long been shrouded in mystery. But what if the key to understanding its origins lies within our own immune system? Recent research has uncovered a fascinating connection between immune cell dysfunction, inflammation, and the development of epilepsy. This groundbreaking study not only sheds light on potential causes but also opens doors to innovative treatments. And this is the part most people miss: the intricate dance between immune cells, inflammatory proteins, and their impact on epilepsy risk. Background and objectives. While epilepsy has traditionally been viewed as a neurological disorder, emerging evidence suggests that immune dysregulation and neuroinflammation play a significant role in its development. However, the exact mechanisms remain elusive. This study aimed to unravel the causal relationship between immune cells, inflammatory proteins, and epilepsy, exploring whether these proteins act as mediators in this complex process. Methods. To tackle this challenge, researchers employed the largest available genome-wide association study, GWAS, data on immune cell phenotypes and inflammatory proteins as potential triggers. They then compared this with epilepsy GWAS data from the FinGen dataset to identify outcomes. 5 distinct Mendelian randomization (MR) methods were applied within a 2-sample MR framework to assess causality. Additionally, a 2-step MR analysis quantified the extent to which inflammatory proteins mediate the effects of immune cells on epilepsy and its subtypes. Results. The findings are eye-opening. 32 immune cell types were linked to epilepsy risk, with 19 increasing the likelihood, e.g., CD19 plus B cells, and 13 offering protection, e.g., regulatory T cell subsets. When examining epilepsy subtypes, 30 immune phenotypes were associated with generalized epilepsy, and 26 with focal epilepsy. Here's where it gets controversial. 8 inflammatory proteins showed suggestive causal effects, with some increasing epilepsy risk, e.g., CC chemokine ligand 23, and others providing protection, e.g., interleukin 13. Mediation analysis revealed that inflammatory proteins account for 6.3 to 13.5% of the immune effects on epilepsy. For instance, CD14 plus CD16 plus monocytes elevate epilepsy risk through increased CC chemokine ligand 23 levels, while effector memory double-negative T cells reduce risk by boosting interleukin 13. Sensitivity analyses confirmed these findings' reliability. Although none of the associations reached Bonferroni-corrected significance, the study strongly implicates B cells, monocytes, regulatory T cells, and cytokines in epilepsy's development, with inflammatory proteins acting as partial mediators. Conclusions. These results significantly advance our understanding of the immune inflammatory pathways involved in epilepsy, highlighting potential therapeutic targets. However, the question remains: could targeting these immune components lead to breakthrough treatments for epilepsy? Future studies must validate these findings across diverse populations and delve deeper into the molecular mechanisms at play. This research not only challenges conventional views but also invites a critical discussion. How can we harness this knowledge to transform epilepsy treatment? Source: Wu, G. et al., 2025. Immune cell phenotypes, inflammatory proteins, and epilepsy. A Mendelian randomization study to investigate causal pathways. Neurosurgical subspecialties. DOID.org/10-14218/NSSS 2025-00010. Suggested reading. For those interested in diving deeper, exploring the role of T cells, https://www.news-medical.net/health/what-a-t-cells.aspx, and the broader implications of immune dysregulation in neurological disorders is highly recommended. As always, while this content is based on edited and approved sources, it's essential to consult medical professionals for personalized advice. Your questions, but not your email details, will be shared with OpenAI and retained for 30 days in accordance with their privacy principles. Please avoid sharing sensitive or confidential information. For full details, read the terms and conditions, https://www.news-medical.net/medical/terms.